Welcome to Med Together. This is a podcast by the people, for the people, as it were. My name is Chana. I'm a med student in New York. This podcast deals with a lot of med school topics, starting from the basic sciences and building its way up, and hopefully it'll be a bit of review that can help y'all consolidate and synthesize important information and maybe phrase it in a way that's easy to understand. By the way, if you're pre-med or you're a nursing student, or you're just interested in sciencey, medically topics, and you're not in healthcare at all, you're totally welcome to. We don't discriminate. If you're interested, you're cool. Ready to rock? Let's do this. Hey everyone, welcome if you're new, welcome back if you're a repeat listener. I'm really excited to be starting this new series on metabolism today. But before I jump in, I do want to express my sincere gratitude to those who've reached out to me via email or in person, for those people who are in the same school as I am, and have told me that they enjoy the podcast or they find it useful. It means so much to me. And I really, really appreciate when you take the time to let me know that it helped you, so please keep it up. And without further ado, let's get right into it. What is metabolism? Metabolism is how our body uses the nutrients that we give it, how they're broken down, and then how they're built back up into the things we need in our bodies. So in order to get a kind of broad overview of what we're going to do in this series, let me just tell you really quickly. We're going to talk about the 4 classes of macromolecules. We'll talk about carbohydrate metabolism. We'll talk about lipid metabolism. We'll talk about protein/amino acid metabolism. And we'll talk a little bit about nucleic acid metabolism as well. Of course, we'll just talk a little bit at the end about what happens when metabolism goes wrong, because that's kind of the interesting part. And I know that hearing about carbohydrate metabolism makes some people break out in a cold sweat and think about glycolysis and the TCA cycle. And most people do not want to try to recreate those biochemical structures. Personally, I thought that those were really fun, and biochem was one of my favorite classes, but I'm not going to subject you to those. The point of this is to try to de-stress it and to highlight the important points you need to remember in order to have a functional understanding of our breakdown of carbohydrates, as opposed to maybe the more granular biochemical understanding you had to memorize in undergrad. I'm also going to throw in a couple of random items that don't really fit into any of those 4 classes strictly, but have to do with metabolism and nutrition also. So we'll talk about some vitamins, not all of them. We'll try to highlight the B vitamins in particular, as well as a couple of other key ones like D and E. We'll talk about iron. We'll talk about some other trace metals briefly, and disorders thereof. We'll talk about hemoglobin synthesis and disorders of hemoglobin synthesis. The way that my med school has always done medical education is to kind of interweave the pathophysiology in with the physiology, and to me that seems like the most logical way to learn it. Like when you learn about how something is supposed to work, it kind of makes sense to then go, well, what happens if it doesn't work? So that's the way I present it in this podcast. All right, now this episode is going to be kind of a short one. We're going to take a 3,000-foot view of metabolism. What are the things that are involved in metabolism? And then a little bit of a reminder and brush up on enzymes. And I know that some of the information in these episodes, particularly this one, is going to be repetitive of things you've learned in the past and maybe even things we've talked about in previous episodes. But I like to quote one of my professors, and I think I even quoted him before in this podcast series. So there you go with the repetition. But he says, everything important will be repeated. And it's so true. Repetition is great for us. The more times you hear something, the more accessible it becomes in your memory, and the more likely you are to remember it on an exam. So for those of you who have eidetic memories, which is that you remember everything you hear, I do apologize for the repetition, but for most of us, I think the repetition is useful. So it's not always completely intentional, but neither is it unintentional when I repeat something that I've said before. All right, so our body is a really pretty well-oiled machine that hopefully, when all goes well, works together in order to convert food into energy, and then uses that energy to carry out appropriate cellular processes. We can think of our metabolism as being of 2 strains. We have anabolic metabolism, which focuses on building, and we have catabolic metabolism, which focuses on breakdown. And these 2 are really irretrievably coupled, because catabolism provides the fuel or the energy for anabolism. And if you're not doing enough catabolism from food intake, like in a state of starvation, you're going to start breaking down your body's reserves of fat and eventually muscle in order to maintain the energy needed for basic processes like maintenance of heart rate, blood pressure, wound healing, and eventually even brain function and consciousness. Now, there is a hierarchy which we'll explore a little bit of which organs get priority when it comes to accessing energy, and the brain and the heart, which are pretty much our vital organs, unsurprisingly top that list. And I also just want to sidestep for a second and say we keep saying energy, so let's remind ourselves what we mean by that. When I say energy, for the most part, I'm talking about ATP. ATP is the currency of the cell. You can have coupling of certain favorable reactions directly with other non-favorable reactions not going through ATP, but for the most part, ATP acts as our main energy carrier source. And how does this work? So ATP is an adenosine triphosphate. It's a molecule of adenosine, with 3 phosphate groups attached to it, and it does not like to have 3 phosphate groups attached to it. So when it's in this state of being ATP, it's in a very high energy state. It's like stressed out. So breaking one of the bonds and leaving ADP, adenosine diphosphate, releases energy because it brings the molecule down to a lower energy state. And that energy, which is released by breaking that bond, can then be used to fuel other energetically unfavorable processes. I will point out that there are other nucleotide triphosphates. You can have GTP as well, but in general, we're talking about ATP, and it's a good kind of way to think about energy as being carried on ATP. And when we talk about metabolism at the whole body level, we're going to be talking about the fasted or the energy poor state and then the fed or energy rich state. And each organ that we're going to kind of break down in this series has its role in the fasted to fed transition. Of course, the spotlight of this series is going to be the liver, because the liver is really the star of the show when it comes to dealing with keeping our blood glucose steady, which is really a huge amount of the focus of our metabolism, is having enough glucose in the blood to supply vital organs like the brain. So like I just said, in the fasted state, the main idea and the focus of our body's metabolic efforts is to maintain blood glucose in order to nourish the brain. We don't want to have to rely on fatty acids, which we can do in starvation states, and sometimes we do, but the brain prefers glucose, and fatty acids have to be transitioned to ketone bodies in order to use them. It's a little messy. It's not energetically efficient. So other tissues are going to spare glucose in a fasted state so that the brain can use it. And the liver is actually going to be actively putting out glucose in this state for the red blood cells, which can't do their own metabolism, and for the brain. So other tissues, when we say they're sparing glucose, what they do is they're using free fatty acids, or they're using their own ketone bodies in order to nourish them and to nourish the liver to allow it to release glucose. Now, this is whole body level metabolism. There is also going to be cell intrinsic regulation that we're going to talk about a little bit, where the cell turns processes on and off depending on its own needs. And then the hormonal regulation is needed to tell the organs what to do in order to enforce a metabolic state for the overall good of the body and get everything to work together as a team. So the big organ that's important in the hormonal regulation of the fasted-fed transition is the pancreas. The pancreas is the major organ that's in charge of this. It secretes both glucagon from its alpha cells and insulin from its beta cells, and these are the two hormones that really control what the liver and what other organs are doing in order to smooth out this fasted-to-fed transition. So in the fasted state, glucagon is the main hormone that's going to be circulating. And the way I remember this, I've always remembered this, is that glucagon gets released when the glucose is gone and you're in a fasted state. So glucagon in this fasted state is mainly going to target the liver. And it's going to tell it to release glucose or to do gluconeogenesis and create more glucose and release it. And it also instructs the adipose tissue to secrete fatty acids, which helps nourish the liver and keep this process going. Now, when you eat something, this information hits the pancreas. The pancreas starts releasing insulin from its beta cells. Now, the insulin does several things, one of which is feeding back on the pancreas and restricting glucagon from the alpha cells, because it would be very disorganized and chaotic and energy inefficient if we had tons of insulin and tons of glucagon in the blood at the same time, because they're sending opposite messages and everything would get confused. So the first thing it's going to do is it's going to restrict the alpha cells so glucagon stops. And then what else does insulin do? It tells the liver to stop doing gluconeogenesis, stop doing glycogenolysis, stop releasing glucose into the blood. It's like, stop, we have enough. Instead, it tells the liver to take up glucose, to try to reduce the blood glucose, to use it for energy, and to replace glycogen stores, which we're going to need in the fasted state. Insulin's also going to target muscle and fat cells and tells them to increase their glucose uptake and to store it. Muscle and fat cells can store glucose as glycogen. They don't release their glycogen and help anybody else, but they do use their own glycogen when blood glucose is low so that the glucose can be reserved for the brain. The liver is unique because it is the only organ that can release glucose when blood glucose is low. Now, weight is a state of homeostasis, where basically if your caloric intake is balanced with your caloric output, you're going to stay the same weight because you're going to be using and storing glucose at the same rate. So it really does come down to calories in versus calories out, although your body will adjust over time if the caloric intake changes significantly. Now, the body does have a bias in terms of this burning versus storing fat. So the body would prefer to store fat because losing fat and being underweight to the point of malnutrition could kill you. So the instinct is to defend your weight. This is what makes trying to lose weight really hard, because your metabolism is trying really, really hard to defend your weight by slowing down and using less energy when you're taking in less energy. overnutrition evolutionally was never a problem, so there's no real check on the weight gain. You actually can gain a whole lot of weight before your body starts to be like, hey, wait, this is a problem. Versus losing weight, when you start to decrease your intake, your body automatically slows the metabolism to defend your energy stores. Now I want to turn our attention to talk about enzymes just for a couple minutes, because all these processes in the body, or most of them, are facilitated by enzymes. And let's just remind ourselves, what is the job of an enzyme? So broadly speaking, in a chemical reaction, the product is going to have a lower free energy than the substrate, but there is a free energy barrier, which is to say that the transition costs energy. And the rate of the reaction is going to be determined by the height of the free energy barrier, the amount of free energy that needs to be put in order to transform it from the substrate to the product. Enzymes are biological catalysts, and their job is to reduce the free energy of the transition state, which we sometimes refer to as reducing the activation energy, and therefore they speed up the reactions. It's important to remember that the end and the beginning free energy do not change. It's just about making the transition state a lower energy state, lowering that free energy hump that has to be overcome in order to get to the product. And we have lots of different kinds of enzymes in our body. Fortunately, the name often describes the function. And some of the names are more straightforward than others, but here are a couple I'm just going to list off for you that are, I think, good to know, because when you come across them later, you don't have to scratch your head and be like, I wonder what that transformation is. You'll know, because if you know the name of the enzyme, you'll know what's happening, and vice versa. If you know what's happening, you can pick out of a herd what the correct enzyme is, because you'll know what kind of function it's supposed to be performing. Let me give you a couple of examples. So the first one, my favorite one, maybe one of the most ubiquitous ones, kinases. These guys catalyze transfer of a phosphate group, usually from ATP to a substrate. Kinases phosphorylate things. We've talked about these before. We'll talk about them again. They come up all the time. Just commit it to memory. I know the word kinase doesn't mean phosphorylate to you necessarily, but it should. Make kinase mean phosphorylate, and it will make your life easier. So then the opposite is the phosphatase, which maybe is a little bit more instinctive. It's the opposite of a kinase. It takes off a phosphate group. Now, unfortunately, we have another enzyme that sounds fairly similar and does something different, which is a phosphorylase. These have a similar end product to kinases. They add an inorganic phosphate, but they do it without the use of ATP. So phosphorylase as a phosphate group, but not with ATP. Dehydrogenases, these you're going to see all the time in redox reactions. They move hydrogens around. Hydroxylases add a hydroxyl group. Carboxylases add a carboxyl group. Mutases, it kind of sounds like mutant. It makes a mutant thing, and it kind of does. These relocate functional groups within a molecule, so they do like rearrangements. And then synthases or synthetases, these use some sort of energy like ATP or acetyl-CoA or something to join molecules together, which, again, the name kind of makes sense. They do synthesis. They're synthases or synthetases. There are other enzymes that I didn't mention in this list. This is completely not exhaustive, but these are just a few that come up fairly often. And it would behoove you to know what these enzymes do, because it's just useful. Now we're going to talk a little bit about enzyme regulation. Why do we need enzyme regulation? So there's a lot of metabolites running around in our body, and we don't want enzymes just on all the time, because we don't always want these metabolites to be transformed. And in particular, the body regulates enzymes that facilitate rate determining steps of reactions. So if we remember what the rate determining step is, I don't know if you remember this from Gen Chem, and it's also kind of logical also, but sometimes a reaction has several steps. It has a bunch of fast steps, and then it has a slow step, and the slow step is the rate determining step, because it doesn't matter how fast the other steps are, they all kind of pile up behind this rate determining step. So regulation of an enzyme that helps a rate determining step is going to be key to regulating the entire process. And that's part of why, by the way, understanding these processes is important, besides for the fact that it's interesting. If you know which are the key steps in these pathways, and we can figure out the enzymes which regulate these key steps, we suddenly have efficient drug targets. So for example, cholesterol has 25 steps in its synthesis, but there's only one rate limiting step, and that's the enzyme that needs to be regulated, and that's the enzyme that gets targeted in a lot of our cholesterol lowering drugs. Now, the rate of an enzyme, the efficiency of an enzyme, is plotted as a Michaelis-Menten curve. This is pretty much like what we talked about in the farm episode with the drug concentrations. It's a similar kind of curve. So you have the Vmax, which is the highest possible reaction velocity, which is analogous to the Emax, the max effect of the drug. Km is going to be the concentration of substrate needed to give you half of Vmax, because enzymes are directly dependent on the amount of substrate. The more substrate you have, the more efficient the enzyme is going to be. And at that concentration, Km, the reaction velocity is going to be half of Vmax, and you're going to get a hyperbolic curve when you plot the reaction velocity as it depends on the substrate concentration. Now we can also linearize this, similar to what we did with the drug curves, and we can make a Lineweaver-Burke plot. And this linearizes Michaelis-Menten somewhat, so you plot 1 over the reaction velocity as a function of 1 over the concentration. The y-intercept is going to be 1 over Vmax, and the slope is going to be Km over Vmax. We use these characteristics of enzymes to talk about what happens when we use inhibitors, which is one way that we can regulate enzymes. We'll talk about a bunch of different ways of regulating them, but one of the most maybe straightforward ways is inhibitors. And we have competitive inhibitors and non-competitive inhibitors. Let's just refresh the difference very quickly. So a competitive inhibitor is a molecule that binds to the active site and it blocks the substrate from binding. So you're just going to basically reduce the throughput through the enzyme. So for example, statins are competitive inhibitors for the enzyme that catalyzes the rate-limiting step of cholesterol synthesis like we talked about. And in this kind of inhibition, the Vmax, which is the max effect, the highest possible reaction velocity, is not going to change because you can outcompete a competitive inhibitor if you flood it with enough substrate. Now, non-competitive inhibitors actually do reduce the Vmax because it binds somewhere else and it changes the enzyme conformation so it can't do its thing or it can't do its thing as effectively. So no matter how much substrate you flood it with, you're never going to reach the maximum possible reaction velocity if you have a non-competitive inhibitor. These ones cannot be outcompeted. Now, another way of regulating enzyme activity, which is a little bit less acute, is regulating enzyme abundance. And this is more like a long-term adaptation thing. So some examples are like bacteria. So bacteria can use lactose for a living. If you take away their glucose, they're going to upregulate the enzyme that metabolizes lactose because they have these regulator genes which will turn on its expression selectively when low glucose is sensed. So this is regulation of their enzymes in response to a change in the environment. It's not going to happen instantly. It's not like flipping a switch, but it's one way of regulating enzyme activity. Another way that we can regulate enzyme activity is kind of an inverse way of thinking about it, but if you regulate substrate abundance, substrate availability, then you're going to change the ability of the enzyme to act because, like we said, the Vmax depends on concentration of the substrate. So if we regulate substrate availability, even if the enzyme is working perfectly, it's not going to do anything. Now, some enzymes are regulated by signal transduction, so they need to be activated downstream of a signaling pathway in order to be active. One example is one that we're already familiar with. We talked about this one, CAMP-dependent protein kinase A, pKa, which is made of a catalytic C subunit and a regulatory R subunit. Just to remind you guys, we talked about this with the GPCRs, but I will go through it again because repetition is great. So the C subunit of this pKa, remember, it's a protein kinase, so what does it do? It phosphorylates things. The C subunit moves the gamma phosphate of an ATP to proteins. That's phosphorylation. Now, the R subunit, the regulatory subunit, blocks the portion where the substrate would bind. So it basically occludes the binding site by a process we call pseudo-substrate inhibition, which is a type of competitive inhibition. Now, cyclic AMP makes the pseudo-substrate region of the R subunit retract. It frees the C subunit, and the free active subunit can affect a range of cellular events by phosphorylating cytoplasmic, nuclear proteins, other enzymes, etc. So this is an example of enzymes being regulated by signal transduction because some kind of signal needed to turn on the production of cyclic AMP in order to free pKa to do its thing. In another example, calcium binds calmodulin, causing a conformational change where it can bind to other kinases and activate them. We saw this in the smooth muscle contraction with the activation of myosin light chain kinase. And again, calcium is released downstream of a signal, and then that calcium binds calmodulin. Calmodulin is the enzyme which can then do its thing because of a signal transduction. There is another way to regulate enzymes which we call proteolytic activation, and this is an irreversible change where the enzyme, which is called a zymogen in the inactive form, is activated by cleavage, by proteases actually. So one example is digestive enzymes in the gut. In the pancreas, these enzymes are produced as chymotrypsin, prolipase, etc. They're produced as zymogens. And then in the gut, there's an enteropeptidase, which is a protease, and it activates these inactive forms by cleaving them to trypsin and lipase and etc. so that they can do their digestive work. And the reason that our body does this is because it doesn't want them to chew up things along the way as they're getting from the pancreas to the gut. And eventually, these kind of enzymes, which are created by proteolytic cleavage, will get inactivated by another cleavage, which makes them inactive. Okay, so there you have it. Short intro to metabolism. Basically, your body's a machine that runs on batteries, mostly in the form of ATP, and there's a complex regulation of several different organs and organ systems to try to balance the production of energy from food with its use to create proteins and carry out cellular functions. Because we're built for survival, we have a biologic basis towards storage and conservation, which is why we put on weight easily and find it hard to lose weight. We also briefly reviewed enzymes, the biological catalysts needed to reduce activation energy of most of our metabolic processes. And we talked about several ways we can regulate our enzymes through inhibition, substrate sequestering, regulation of enzyme abundance, and enzymes existing in forms that need activation via another molecule or even via proteolytic cleavage. Thank you so much for listening. Our next episode will start with tackling carbohydrate metabolism, hopefully relatively painlessly. But for now, I hope you enjoyed, and if you did, please consider taking a moment to rate the show and mention it to your friends and fellow students. I'd really appreciate it. You can feel free to reach out to me at any time with questions, comments, or requests. My email is medtogether26@gmail.com, and I'd be delighted to hear from all of you. Catch you in the next episode.